Hello and welcome. My name is Dayanara, and today we're going to be talking about cells, organelles, what's in between the cells, and a special type of transport. So, overview. Specifically, we're going to be talking about the rough endoplasmic reticulum, the ER, the Golgi apparatus, vacuoles, extracellular matrix, and the special type of transport, primary active transport. Starting off, the rough endoplasmic reticulum. The function of the rough endoplasmic reticulum helps make proteins and prepares them for use in the cell. It does more than just make proteins, it also helps fold them into the right shapes and adds certain chemical groups and sorts them where they need to go. It is essential for producing proteins needed by the cell and for export. It plays a critical role in making sure these proteins are folded correctly and chemically modified. Errors in this process can lead to cell stress or disease, and the cell won't function. As you can see in the picture, the rough ER is this folded membrane cheese-looking stuff right next to the nucleus, and like your favorite Valentino bag, it is studded, but instead of rhinestones, it's ribosomes. They are constantly attaching and detaching from the ER membrane as needed when making proteins. It is only found in plant and animal eukaryotic cells because only eukaryotic cells have membrane-bound organelles, which the rough ER is one of them. Prokaryotic cells lack membrane-bound organelles. Next up, the Golgi apparatus. The Golgi apparatus processes, modifies, and sorts proteins and lipids received from the endoplasmic endoplasmic reticulum, the one we just talked about. It packages them for delivery to their correct destinations, such as the plasma membrane, lysosomes, or for secretion. It is essential for proper protein and lipid modification and trafficking. It ensures proteins are chemically altered for their final roles and directed to the correct locations. Inaccurate Golgi function can lead to misfolded or misrouted proteins affecting the cell health. It is in both animal and plant cells. The Golgi modifies and ships proteins and lipids. However, in plant cells, the Golgi also synthesizes complex polysaccharides for the cell wall. In yeasts and plants, which do not have lysosomes, the Golgi helps send proteins to the vacuole, which we'll talk about in a second. In the picture, it's right here, it kind of looks like cheese as well. It has a ribbon shape, just like the ER. It is made up of a series of membrane-bound sacs called cisternae. It's these right here. Each one plays a different role in processing. The lumen is the inside space of the cisternae, or a single one is a cisterna. It's where the chemical modifications happen. It's like the little light yellow inside bit. Next up, vacuoles. Vacuoles store and manage substances like water, waste, toxins. They keep the cell clean by collecting waste and sometimes breaking it down, similar to lysosomes. They protect the cell and keep its internal environment stable. In plant cells, there is one large central vacuole, like in the picture here. It takes up more than 80% of the cell. It is essential for storage, support, and protection. The vacuole stores water, salts, nutrients, proteins, pigments, and waste. By filling with water, it creates something called turgor pressure, which helps the plant stay firm and upright. When the water is low, turgor pressure drops, causing the plant to wilt. So when you're not watering your plants, this is why it wilts. In contrast, animal cells have smaller vacuoles used to store or move materials for short periods. Onto the extracellular matrix. Say that 3 times fast. The ECM, extracellular matrix, provides structural support and organization to tissues. It acts as a scaffold that holds cells in place and helps them stick together. So technically it's not an organelle, but it's like in between the cells in a tissue. It also sends biochemical and mechanical signals that affect growth, movement, and survival. It helps regulate water balance, stores growth factors, and allows tissues to resist stretching or compression. The ECM is essential for development, repair, and tissue health. It helps form tissues, keeping cells functioning, and responds to stress or injury. Damage in ECM proteins can lead to diseases like fibrosis and cancer. The ECM is found in animal cells where it fills the space between cells. Plant cells don't have ECM, they use a cell wall. ECM is also absent in prokaryotic cells. It changes depending on tissue type and whether it's healthy, injured, or cancerous. In the picture, you can see the plasma membrane of a cell. Above it is the matrix. The thick tan fibers are collagen fibers for structure. The gray-blue fibers are proteoglycans, made of carbohydrates and proteins and polysaccharides. They attract water and act as cushions. Fibronectin is the matrix protein that helps other proteins attach, keeping the ECM organized. Primary active transport. Primary active transport is when cells use ATP energy to move molecules against the concentration gradient from a low to high concentration. This is done with transmembrane proteins called pumps. These pumps use ATP to change shape and move ions, like 3 sodium ions out and 2 potassium in. It's crucial for maintaining homeostasis, especially ion balances like sodium, potassium, and hydrogen. Disruption can cause diseases like cystic fibrosis and kidney disorders. It happens in many cells. Sodium-potassium pumps help maintain membrane potential neurons, hydrogen pumps control pH in kidney cells, and calcium pumps help with muscle contraction and heart signaling. It helps with cell communication by maintaining cell potentials, which are needed for neuronal action potentials. The sodium-potassium pump is required to make these potentials, letting neurons send electrical signals. Alright, questions. Which of the following describes the function of the Golgi apparatus? A. Produces energy for the cell. B. Synthesizes DNA. C. Modifies and stores proteins and lipids. Or D. Stores genetic material. 2. Primary active transport uses ATP as a source of energy to move molecules blank their concentration gradient. 3. How does the sodium-potassium pump support cell communication? By creating glucose channels, by creating ATP for protein synthesis, by maintaining electrical gradients needed for nerve signals, or by breaking down cell waste. I await your answers. And here are my references. I have a lot of them. Thank you for listening, and I hope you found this educational.